G'day, I'm Dr. Philip Button from the Food Microbiology Academy. Here I am in one of the old style lecture theatres on the Clayton campus of Monash University. This is in the S14 lecture theatre within the science precinct, so within the Faculty of Science. And this is one of the old style lecture theatres where you can see behind me, it's their blackboards. So this is pretty original, going back quite a number of years. What I want to do in this session is take you through a lecture, few introductory slides on pH, on a lecture that I conducted several years ago titled Intrinsic Factors Influencing Microbial Growth in Food. So to start with, what are the factors that do influence microbial growth in food? Well, basically there's two factors. Intrinsic, those that are intrinsic, and those that are extrinsic. So when we're talking about intrinsic, those are the ones that are within food. So these are the ones that are naturally part of the food. pH, water activity, nutrient availability, and the presence of any inhibitory substances. So these are the materials, these are the ingredients, these are actually part of the food, the parameters, the properties of the food itself. Then we've got those factors that are external to food, and those are the extrinsic ones. So one of the best examples of those is temperature, and atmospheric conditions is another one. So whether you've got modified atmosphere of oxygen or some other type of gas. So for the next, the rest of this session, what I want to take you through is about pH, review of pH to start with, and then some key elements of pH as it pertains to particular types of food, food groups. So what does pH actually mean? So pH actually refers to the power or potential of hydrogen, and it is related to the self-ionization of water. So that represents the acidity or the alkalinity of an aqueous solution, and is the hydronium or the hydrogen ion concentration on a log scale. So that's a really important element to remember, that it's a log scale, so that pH of 5, for example, is 10 times more acidic than a pH of 6. So it's not a linear scale, it's a log scale. Now, pH is a really important factor in controlling microbial growth. And most foods are actually either acidic or neutral. And the pH preference of bacteria and fungi is actually, well, as far as bacteria go, most bacteria prefer a pH that's around, that's in the neutral range, whereas fungi tend to do better in the range that is slightly acidic. And with pH, you can have organic or inorganic acids, which make which can create the acidic environment. So we're talking about, if we're talking about organic acids, well, then it's like acetic acid or lactic acid, whereas inorganic can be like sulfuric or hydrochloric acid. And an important element to note is that the spoilage of acid foods takes longer than the spoilage of foods at neutral pH. And really, that is all because of the types of microorganisms that prefer those particular growth environments. So as I mentioned before, we've got bacteria that prefer that grow optimally within the neutral pH range. So, 6.5 to 7.5, so around 7. And bacteria grow much faster than fungi, and therefore, if food is in that neutral range, then they're going to be spoiled quite fast, because bacteria grow faster, and that's the optimal temperature. That's the optimal pH for bacteria. Whereas if the pH is in the acidic range, so 4, 5, even up to 6, for example, then those are the pH values which are going to be spoiled by fungi predominantly, because those pH values are going to be inhibitory to bacterial growth. So as a result, they favour the growth of fungi, and therefore they're going to be spoiled much slower, because fungi will grow much slower. So that's quite an important distinction to remember about pH, the type of foods and the type of microorganisms that you'll get spoiling those particular types of food based on their pH value. So if we turn to some specific types of food products, so fresh produce, for example. So if we start off with fruits, for example, a lot of fruits tend to be in the pH range of 2.5 to 4.5, thereabouts. So they are they can be very acidic indeed. So much too acidic for bacterial spoilage, bacterial growth to occur. And the common acids in fruits are, for example, malic acid in apples, citric acid in citrus fruits, and we've got tartaric acid in grapes and mangoes. And it's of course, the higher concentration of these particular acids which will, have these low pH values, which provide really perfect conditions for growth of fungi. So you can see that, if you think, you can see that fruits predominantly spoil because of growth of fungi. If you think about, pears or citrus fruits, they're likely to succumb to growth of mould, for example, yeasts and moulds due to these low pH values, which inhibit the growth of bacteria. So with moving on to vegetables tend to be moderate to low acid. So from around 4 to 6.5 pH value, and the vegetables tend to be poorly buffered, and so this allows for a succession of organisms to actually develop. So one of these is the growth of sauerkraut, for example, where you have one particular type of microorganism which starts the fermentation process in sauerkraut, which changes the pH value because vegetables have a low buffering capacity. Then this leads to another type of, another community of microorganisms developing because the pH value is too low for the previous ones to actually continue their growth. So with the spoilage organisms in vegetables, though, because of the higher pH, it's much more common for bacterial spoilage to occur. Of course, if it's around 4 to 5, in that sort of range, then fungi are more likely to proliferate. But especially when we approach 5.5 and getting above 6, then we're certainly getting into the range where bacteria are likely to grow well. And even if they're present along with fungi, they're going to grow faster and therefore outcompete the fungi in those particular environments. So especially Pseudomonas, and various other soil-dwelling plant pathogenic microorganisms as well, or those that are related, can take a foothold. And when I say take a foothold, that's particularly so when barriers are broken. So we're talking about entry being given to these particular bacteria as a result of the peel or the skin being compromised in fruits, for example, tomatoes, zucchinis, and so on. Fruits and vegetables. So what we want to talk about now is meat. And meat tends to have pH of around about 6. And in the case of raw, chilled, aerobic, stored meat. And especially for export markets and those sort of things, we have Pseudomonas. That tends to be an issue, and we tend to have, in terms of the sensory outcomes from this sort of spoilage, it's much more related to protein, and protein, the proteases that are produced by these bacteria, and the putrefaction, the degradation of the proteins, and hence the spoilage, which is protein related. So we also have Pseudomonas if we're talking about psychotrophic organisms, or we could have Psychobacter if we're talking about psychophilic. But the interesting thing is that many microenvironments actually develop, or are present on a meat product. So we're talking about, on the surface, and even inside to some extent of the meat product. So by and large, what will happen on the surface is that the surface is exposed, and that's where you'll get aerobic microorganisms growing. But if somehow the meat inside the meat is more likely to be sterile, or should be sterile, right? But under some circumstances, you can get microorganisms developing inside, in the interior part of the meat. And these are likely to be anaerobic, though, because of the lack of oxygen. And these contaminants may grow and deplete the oxygen adjacent to bone, creating an anoxic habitat, which is a deep bone spoilage by anaerobes. So the last two slides. The last two slides are actually on pH and its relationship to total titratable acidity. The last two slides in their series. So what we're going to actually do is cover these in another video, because these are pretty, getting into basic elements of chemistry. So we're going to skip those for now, and we're going to have those as a separate session on the chemistry of pH. So please look out for that video, and in the meantime, take care, all the best, and goodbye from one of the lecture theatres of yesteryear here on the Monash University Clayton campus. See you around.